This is a tape on chemical. The human eye is the best instrument and the most effective tool for chemical identification in the whole wide world. Every day you and I identify thousands of objects just by eye. A robin, a banana, a dollar bill, a door key, a ballpoint pen, any of thousands of objects. We look at and recognize instantly a water glass. Well, that's a mixture of oxides of silica, sodium, calcium, and potassium. So you've made a chemical analysis. You pick up a fork. That's iron plus silver coating. You eat your scrambled eggs, a mixture of amino acids. You add more salt, sodium chloride, and butter, a mixture of proteins, mostly casein, carbohydrates, fatty acids, vitamins, and water, H2O. The bar of soap we use for our shower is a mixture of sodium salts of fatty acids, principally palmitic and oleic. And so the day goes on. You recognize thousands of objects during the day, all of known composition. So we, proteins plus a tricalcium phosphate skeleton, are each a walking, talking chemical analytical laboratory. The microscope simply magnifies objects too small for us to see to a larger recognizable size, like these radiolarians, silica, SiO2, little marine protozoa. This is a pollen grain, pine pollen. You might be able to see, if you look carefully, why we call it the Mickey Mouse pollen. All pollen grains are different in size, shape, color, and we can identify the species of the plant just by recognizing these particles of pollen. This is ragweed pollen. Ragweed pollen is obviously different in shape. I call it the bottle cap pollen to help me recognize and remember it. We'll just see a couple of these pollens before we switch to leaf hairs. These are hairs that grow on the underside of leaves and grasses, and they are distinctive with respect to the species of the tree or the plant on which they grow. They're called stellate hairs, and these come from the sycamore tree. The underside of the leaf is really felty from these hairs, thousands of them. These are salt crystals, sodium chloride, right out of the salt shaker on the dinner table. They are little cubes, distinctive in shape and optical properties. If we needed to measure them, we don't, to recognize them, especially when we shake them out of a salt shaker. These are sugar crystals, right out of the sugar bowl. Different shape. They're not little cubes. They're more complex in shape, have angles other than 90 degrees. And again, they're recognizable as sucrose, sorry. These are potato starch grains. Potato starch is a nice, smooth, round, clear, colorless particle, cellulose. It's different in shape from starch grains of rice or wheat or cornstarch. These are cornstarch. They're more or less subangular in shape because they grow in a compressed mass of them that press in contact with their neighbors. These are fibers. They're paper fibers from a tree, wood fibers, softwood fibers. Recognizable as such by a microscopist. They're little flat ribbons. They have little pits through the walls of the ribbon, which we don't see here very well. These are also cellulose, like the wood fibers. These are cotton fibers. They're not ribbons. They're twisted. And the twists are what tell us that this has to be cotton. There are many natural fibers, all of them identified by what you see looking at them microscopically. The next ones that we look at are wool. All animal hairs have scales, and you can see the scales very nicely on sheep's wool here. Even our hair, human hair, has scales. After all, we're animals. And the ones that we see here are my hair. A long time ago, this is an old standard slide of mine that I keep. It's about 40 years old, before I had gray hair. These were nice brown hairs. The scales aren't as easy to see, but the sort of cross-hatching on them are the scales. They are easily differentiated from most other animals, like rabbit. These are rabbit fibers. The little ones, two little ones, are very fine fibers. Most of the fur is made up of those very fine fibers, but the guard hair, or the kemp down the lower left, is stiff and straight and keeps the fur fluffy, so it's a good insulator. These are synthetic fibers, viscose rayon fibers. They have longitudinal striations, and that characteristic cross-section helps to tell me immediately that they are viscose rayon. All synthetic fibers have characteristic cross-sections. These are nylon fibers, perfect cylinders. They are smooth-walled, they're clear. These particular ones are colorless. They could be dyed, of course. These are also wool, but these are mineral wool, so they're glassy. They're practically the same composition as window glass. And they're silicates of calcium, sodium, potassium, magnesium, aluminum, so on. But they have different diameters, and they're not straight. So they're differently formed and easily recognized. These are asbestos fibers, chrysotile asbestos, easily differentiated from anything else, and including 5 other different types of asbestos. Obviously, these are recognizable at sight if you've seen them before and if you have a good memory. These are combustion products. They are pulverized fuel boiler fly ash from a electricity generating power plant. They're completely burned, little glassy spheres of the molten and then solidified ash. There are other things that we can do under the microscope to help us recognize things more rapidly. These colored crystals are a colorless mineral olivine, but that we've treated in an optical manner to give these colors which depend on their orientation and thickness. This is called dispersion staining, and the mineral is called olivine. We can recrystallize compounds like the salt and sugar from water on a drop on a microscope slide, and the crystals that form then are characteristic of that particular substance. We can measure angles if we need to. We can measure refractive indices and so on. These are going to be crystals of a metal itself. Lead, in fact. We have here crystals in the top of metallic zinc. And they, because they're higher in the electromotive series, more active than lead, will displace lead from solution. And you can now see crystals of lead actually growing in the droplet of water. The spewing that you see there, the flume, is lead carbonate, which is forming from the lead displaced from solution, reacting with the carbonate, the CO2, that dissolves in any droplet of water, since there's CO2 in the air. So here we see more dendrites of metallic lead. We can do this with copper, with silver, a number of different metals. We do it with mercury, we get little droplets of mercury instead of the dendrites, since it's a liquid material. We're now recrystallizing lead nitrate from water. Again, a tiny drop. The edge of the drop is along the left. We're going down the inside the droplet of water where we have the crystals that have crystallized out as it evaporated. The bigger crystals are ones that I put in and crushed up a little bit in order to saturate the droplet of water. But now as it cools and evaporates, the crystals of the soluble compound lead nitrate are crystallizing out, and we can see their shapes, measure their angles, and we use this as a way of getting well-formed crystals of a given compound in order to recognize it. There's a beautiful little octahedron sitting in the center of the field. This is a chemical reaction between zinc ions in solution and a reagent, potassium mercuric thiocyanate. We see druzy crosses here. These druzy crosses are characteristic of the presence of zinc in the solution. This is a similar reaction with the same reagent looking for copper. And the crosses that we now see are actually the zinc crosses that contaminate this particular solution. But as we continue to look, we'll see what look like sea urchins. These sea urchins, or a group of them here, are characteristic of copper when precipitated with the same potassium mercuric thiocyanate reagent. It's supposed to form little propellers. They're pointed rods that have a hub in the middle, looking very much like a propeller. They'll come out in a few more seconds at least and follow these sea urchins. I just saw a couple of them, but we passed by them a little bit too quickly. There's two of them, big ones. There will be a number of smaller ones. There's some in the lower left corner now. And here's some in the upper corner, upper right, left. And there we are again. They will thicken in the middle and become sort of diamond-shaped before they're through. But this is TNT crystallized from the melt under a cover slip. And that was the low temperature form. This is the high temperature crystal habit of TNT. It's growing just below the freezing point. This squiggly crystal front is very characteristic of TNT. Makes it easily recognizable at sight. Many compounds can be identified this way by looking at the characteristic crystals that form after you melt them and cool the slide so that they crystallize on cooling. This is the supercooled melt of cholesterol acetate, and it's now crystallizing, and we'll see in a second the liquid crystalline form nucleate. There it comes, and it's grown kind of smoky, and it grows across the field very rapidly. It's a liquid crystalline phase. It's a polymorphic form of cholesterol acetate, and it's has no backbone. It flows more or less like a liquid. This is the solid phase of cholesterol acetate growing through the liquid crystalline phase. As they cool, the crystal habit changes and they become prettier as spherulites. They're a good example of the spherulitic formation of millions of tiny little needles radiating out from the center, and still liquid crystalline phase flowing there as it's crystallized into the more stable solid phase of cholesterol acetate. If we look around a little bit, we'll see different thicknesses of the preparation, and the spherulites will change color. That's a nice set of colors there. And we have to rotate the stage when we ever see things like this. Pinwheel effect. This is DDT. DDT grows very slowly, especially at room temperature. It would take a number of hours actually for this whole preparation to crystallize. It starts out growing as a single needle that bifurcates on each end and thickens on those ends, producing something that looks a little bit like a bow tie. There's a little one there. These bow ties then gradually get bigger and broaden and form the spherulites that we see. We'll look for a reasonable spherulite to watch for a minute to see how it fully develops and begins to grow as a circular spherulite. Few there, but that's a little better. We might see that's a pretty good one. I think we might use this one as an example. This one is a good example of a spherulite almost completely developed. I think this is a good place to stop with this tape. Thank you very much. Here we've had a